Liz Noel is a genetics PhD student who comes to the Whirling Lab from the Dominican Republic via New York City. Liz completed her bachelor's degree at the City University of New York, Brooklyn College, after which she worked as a research tech in Suzanne Leal's lab at Columbia University, where she got to dive into human genetics and study genetic variants that are responsible for Mendelian disorders like non-syndromic hearing impairment and intellectual disability. And Liz's work at Columbia earned her authorship on 14 publications, which is quite a feat. Liz's PhD work has taken a bit of a shift from that focus. She focuses on a mouse model of immune responses in pregnant mothers, which is itself a risk factor for developmental disorders, including autism and schizophrenia, and she'll tell you a little bit more about that in her talk. For her project, Liz was the one in the lab to establish several key protocols for us, including behavioral testing and microdissections of mouse brain. And she's an integral contributor to any of our projects involving mice in any capacity. And also, as part of that work, she's collected a lot of behavioral data. And as some in the room may know, scoring behavioral data from animals can take many hours and requires independent observers to do it correctly. And so Liz has found herself mentoring a team of undergraduates throughout the lab over time to assist with this work and other wet lab tasks. And she's been a very attentive and a great mentor and a good leader for those undergraduates. And they have contributed to her work in some substantive ways, which I think she'll point out today as well. And more recently, Liz has also been stretching her comfort zone to learn how to analyze the RNA-seq data that she has generated, which is part of what she'll present for you today. So today, Liz will share her work in progress about her project investigating how a mother's immune response affects the behavior and transcriptomic profile in her offspring. Welcome, Liz.
Hello, everyone. You can hear me, right? So, like Dana said, my work focuses on understanding how maternal immune activation and sex differential biology can affect brain development and functioning. So I am part of the Whirling Lab with Dr. Whirling, and then we have Sean, who's our data scientist. We have Kyle and Roy, who are also graduate students in the lab. Kevin, who is our research assistant, myself, and then Matthew, who's an undergrad. I decided to add Matthew there because Matthew has been working with me since I started my PhD, since he was a freshman, so almost 5 years together. So what do we focus on in the lab? Our main goal is to understand how genetic variation and sex differential biology influence risk for neuropsychiatric conditions. And why are we interested in this? Well, neuropsychiatric conditions shows sex bias in diagnosis. And this is all kinds of neuropsychiatric disorders, from neurodevelopmental disorders like autism and ADHD and schizophrenia, all the way down to neuropsychiatric disorders like Alzheimer's and Parkinson's disease. In lab, we focus mainly in autism, which has a sex disparity of 4:1. So for every 4 males that get diagnosed with autism, one female gets diagnosed with autism. So what is autism? Autism is a neurological and developmental disorder. It affects how people interact with others, communicate, learn, and behave. And symptoms generally appear in the first 2 years of life. So kids most likely get diagnosed after they turn 2. When we think about autism, I think we all like to think about autism as a linear spectrum, when in reality, autism is more like a variable wheel, because every person that has autism showcases it differently, right? So someone might have, really dense problems with emotion regulation, but not so much with speech, where someone else might have problems with eye contact, but not with anxiety. So autism spectrum disorder is more like a variable wheel rather than just a linear disorder. Over time and place, this sex disparity that we observe in autism stays the same. So in this figure here, I'm showcasing you data from the CDC all the way down from 2010 to 2020. And this is prevalence per 1,000 children. Blue bars signify males, red bars signify females. And as you can see, although autism diagnoses are increasing throughout time, that sex disparity is still the same. Here in this figure, it looks into the prevalence of autism in all the states of the United States. Again, red signifies females, blue signifies male, and you can see that also the location really doesn't matter. That disparity stays the same. There's multiple reasons we believe this is happening. One of the ones that we believe is one of the main reasons is maternal infection, and maternal infections have been associated with increased risk of developmental disorders in children if they happen during pregnancy. So for the first table, it's just a paper that looked into schizophrenia. And as you can see, the odds ratio of schizophrenia goes up if the mother gets diagnosed with one of these viral infection during pregnancy. In the second paper, they look into the diagnosis of autism per maternal infection diagnosed during an inpatient hospitalization. And as you can see, the ratio for autism diagnosis, it's also higher than that of control. Okay. One of the possible mechanisms that we believe is creating this is a maternal immune activation. And this happens when there's a viral infection during pregnancy. Um, sorry, I can barely see here. There's a viral infection during pregnancy, and the moms get sick, and then this immune activation creates cytokine signaling cascades to respond to it. And all these molecules that are created by these cascades cross the placenta and they reach the fetal brain. And this leads to long-term postnatal effects that affect health, ability, neurodevelopment, behavior, communication, and a set of other behavior. Now, the MIA connection to neurodevelopmental disorder, it's not new. The first initial hypothesis was made back in the late 1990s, 2000s, but the hypothesis was that if you get sick while you're pregnant, the virus is gonna get to your fetus. When in reality, in 2003, it was proven that actually that's not what it's happening. What it's happening is that if you get sick during pregnancy, the molecules created by the immune system is what's reaching the fetus. Later on in 2007, the broad link was made to neurodevelopmental disorders. 2008, we saw they proved altered gene expressions. 2012, 2015, now we have concrete data, human data that associates it with schizophrenia. In 2017, we find that the risk increases in pregnancy infection. 2018, we find sex differential effects. In 2023, I wish, 25, which is now, is the ongoing research. And this is where I come in and our lab comes in. And this is where we're trying to find out how sex differential effects of MIA stem from gonadal or chromosomal complements. So these are my aims. So aim one is to identify offspring behavior impacted by maternal immune activation and sex. And our expectation was that MIA offsprings would showcase more anxiety-like behavior and would have less social recognition and social behavior. And this would affect males at a higher rate. For our second aim, we wanted to quantify transcriptomic effects of maternal immune activation and sex in offspring's brain. Sorry. And our expectation was that the offspring that were exposed to maternal inactivation will have changes in their gene expression involved with function of macroglias and astrocytes. This figure here is just showcasing how deep each aim is going to help us answer questions. So behavioral assay is going to ask us if there is any change, by MIA, whereas the RNA-seq is going to help us answer if there is any brain regions are being affected, any cell types are being affected, and any genes are being affected. Oh, okay. So how does MIA affect wild-type female behavior, male and female behavior, and brain gene expression? So this is just a little outline of my project. Pretty easy, starts with mating animals, wild-type male. And at embryonic day 12.5, we give the pregnant dawns an IP injection of poly-IC, which is a chemical that mimics a viral infection. Then we move on to behavioral assays that start at postnatal day 3. And then after postnatal day 12, the animals take a big chunk of rest, and then they start behavioral assays again once they turn 60 days old. And after we're done with running behavioral tests, we dissect their brains and we analyze data. So, so I'm going to start with showcasing some behavioral findings that we have on this project. The main questions that we wanted to answer when we were doing behavioral assays were: one, how does MIA affect gene expression, treatment effect? Are sex differences in gene expression altered by MIA? And our brain region? And these are not the questions. The questions we wanted to answer were: is MIA affecting the behavior of the animals? And are the animals showcasing autism-like behavior? This is later. So these are a group, and these are the N number that we have for each group. And all of this animal went through behavioral assays, so all of them were tested. And these are the behavioral assays we ran, the purpose and the rationale as to why we did them. And now, if I was to show you every assay, the results of every assays that we ran, we'll be here for like the next week. So I just put the things that were significant in a little table to summarize. So for neonatal vocalization, where we check for USB count, so seeing how many calls the mice made when they were tiny little pups, neonates, we found that there is a treatment effect, and treatment animals made less calls in comparison to control animal. When we look at olfactory preference, when we look into olfactory preference, in the stage where the mice were presented a non-social scent versus their same-sex scent, we found that female prefer the non-social scent instead of the same-sex scent. In open field, where we test for motor behavior and anxiety, we actually found that males spend more time in the center square. So think about the open field as just like a big square box. Typical mouse behavior, they want to stay within the corners. Our mice like the center more. Why? We really do not know. For the elevated plus maze, where we look for anxiety behavior, we found that the females had more entries into the closed arms. And then for the 3-chamber, which is social recognition and like social interaction, we found that females did not like to, would prefer to interact with the non-social stimulus rather than with the social stimulus. And then for unrestricted social interaction, this is where we had our mice interact with the stimulus animals without any restraint. I put a star because everything was significant, but everything was significant as expected, meaning our female animals prefer to be with female animals. our male animal preferred to be with female stimulus. Our male-to-male interactions were a little bit more rough, whereas our female-to-female interaction were not rough. So everything was significant, but it was as expected. So next, I'm going to show you some raw data just for 2 assays. Do not get scared. So the first one is the treatment effects for olfactory preference in animals. And this is the stage where we introduce 2 Q-tips. One Q-tip had a non-social scent, so this was either lemon or peppermint, and then the other Q-tip had their same-sex scent. This was either female or male, depending the sex of the animal that was being tested. And as you can see, we start with a control difference, a sex difference in control, where females control prefer to smell the same sex scent in comparison to control males. Whereas in treatment, we also observe a sex difference, but it's flipped. Now we have treatment males prefer to have the same treatment males prefer to investigate the same sex scent rather than non-social scent. And overall, as you can see, treatment females were the ones that were affected the most. They really did not care much for that same sex scent. They prefer that non-social scent in comparison to all the other groups. Because we saw this difference, the next question is, well, is this a real effect, right? Is it just because they don't want to investigate, they're tired, they're bored, they don't really care because it's just a female scent? So we did the next thing. We introduced a same-sex scent, Q-tip, versus a opposite same-sex scent. And as you can see, treatment affects olfactory preference in animals, but only in certain environments. Because once you introduce those 2 social scent, that effect that we saw before goes away. This one is the social preference index that we gather from the 3-chamber. So the 3-chamber analysis is a triangle and is divided into 3 different chambers. The 2 outer chamber have 2 cells where you can introduce stimulus animal. So this one that I'm showcasing you, the social versus non-social, was the one where we introduced a stimulus female animal in one of the cages, over-optimized female animal in one of the cages, and left the other cage alone. And then we calculated the social preference for each animal to give them a number. And as you can see, there is a significant difference between control males and control females, but overall, treatment females had more animals with negative ISP. And to check that this is actually a real result, we look at the percentage. So treatment females had 27.3% negative ISP. Treatment males had 15.8%. Control females had 11.8%, and control males had 5.9%. There is some type of treatment effect. So what's next? We introduce another stimulus animal, right? Because now we want to know, do you want to socialize with the animal that you already know, or do you want to socialize with a new animal? It goes away. That big difference that we saw goes away, right? So now you have 50% of treatment females. They would rather socialize with the old animal rather than the new animal. 36.8 treatment males, 35.3 control males, 52.9 control females. So, what are the take-homes from these behavioral assays? Well, I think the main take-home point that we can make about this is that behavioral assays reveal some treatment and sex-related differences. However, the effects were modest, right? and they do not provide sufficient evidence for us to support autism-like behavior. Next, they also they are messy. They are super, super messy. Those results that I showed you felt like whiplash because that's what I wanted you to feel like. You felt like you found something and then you didn't find anything. But it's what I wanted you to feel because behavioral assay, it's like that. And that is where collaboration work comes in. Where is Martha so she can hear my R&R course point? This is where collaborative work comes in. When Donna and I were looking at those results, we didn't know what to make of them. In reality, we were like, well, a lot of time that was wasted. But when we went to someone in my community that had a lot more experience than us looking into behavior work, she was actually a lot more positive about the results than we were. And although those results are not super strong when we can say our animals are showcasing autism-like behavior, we can at least say that at some point, treatment is affected some of their behaviors, right? Yep. Next, I'm gonna go into genomic findings. Okay, this is where it's supposed to go. So the primary research question that we had when we were doing genomic work was one, how does MIA affect gene expression? Two, are sex differences in gene expression altered by MIA? And are brain region differences in gene expression altered by MIA? So for this work, we had three regions of interest: the medial prefrontal cortex, the medial preoptic area, and the BMHBL. Why? The medial prefrontal cortex is associated with social and interactions behavior. Medial preoptic area is associated with mating and parental behavior, and BMHBL is associated with aggression and mating behavior. For these results, I'm only going to be speaking about the MPOA and BMHBL. So what does, truthfully, the MPOA and BMHBL look like? This is from the Allen Brain Atlas, and this is what the MPOA and BMHBL looks like. So this is your MPOA and your BMHBL. But I'm going to brag a little bit because in reality, this is what a mouse brain looks in comparison to a rat brain. And this here's your MPOA. This is a comparison to ferret and a monkey and a human. So we're conducting micro dissections of these brain regions in little tiny, tiny, tiny mouse brains, right? Especially the BMHBL. So we have to use specialized tools to make sure that we are collecting the correct tissue, because if we don't, then everything is going to be messed up. This is just a workflow of the transcriptomic analysis. Nothing super important. And the comparison we did when looking into gene expression changes are sample size. These are the brains of those animals that went through behavior. The starting end looks smaller because we did have to pool animals. So each sample was 2 to 3 animals pulled together because those brain tissue are so small. And then this is just a table looking into the DEG distribution. And these are the comparisons here of the MPOA, BMHBL, the genes that were upregulated and the genes that were downregulated. As you can see, we found a lot of sex differences, but we did not find a lot of treatment differences. So I would only be speaking about the sex differences that we found and not the treatment differences. So to start with the first question, how does MIA affect gene expression? Well, we found that there was minimal impact in adult MPOA and BMHBL. Next, are sex difference in gene expression altered by MIA? So here we're looking first at the MPOA. And when we look at the MPOA, we found that poly-IC introduces new sex dependence changes in the MPOA. Oh, where's here? Okay, so this figure, you're looking at the sex CGs of female versus male control in the MPOA. Your x-axis is your x-axis is your log2 fold change of male versus female, and your y-axis is just p-value. And then this is poly-IC, and as you can see, in poly-IC, there's a lot more DGs, and you can see here also the DG counts increases. When we look to see if there was anything interesting about these genes by doing an enrichment analysis, we found that there were some staff that were interested in regards to the work we do. So we found that, why is this small? Okay. So we found that neuroactive ligand receptor and hormone signaling is female skewed in control. But when we look into poly-IC, those two terms become male skewed. And this is really important for us because one, this can be triggered into the sex difference that we're seeing, but also neuroactive ligand receptor is important for us because it has to do a lot with neurodevelopment. And we're looking at a disorder that has to do with neurodevelopment. Next, I'm just gonna, because no, it's small. Next, we're looking at the MPOA sex differences in gene expression, males versus females, without sex chromosome. I decided to just put autosomes here, so the differences are clear, more clear. In your x-axis, you're looking at your control sex versus females log2 fold change. In your y, you have your poly-IC log2 fold change. All right. And no, sorry, guys. There's a problem. Where did he put it?
What are you trying to do?
The real slide that I had on, the ones where I had made the edits already.
I don't know.
I don't know where he put it. That's why I'm like, what's going
If
on?
this is a different file open?
Yeah, I don't know. Put it. Okay. Just don't mind the regression line here. Something is wrong with the file, so please don't mind the regression line. But what we saw, so here the green dots represent the genes that were affected only in poly-IC, that were significant only in poly-IC. Purple show the genes that were significant only in control, and orange show the genes that were significant for both groups. And as you can see, there's kind of a shift when it comes to poly-IC. There's a flip-flop, what I call like a flip-flop effect. And you can see that here in the heat map, where when you look into control, you see that females and males are different. The expression of these genes are different for females and male. But when you introduce poly-IC, that difference goes away. Whereas for these genes, there's not a difference under control between females and males. But once you introduce treatment, there's a difference, a sex difference comes up. Okay. So moving on to the VMHBL, we found that stronger treatment-induced shift in sex bias GEs were found in the VMHBL. And again, this is just showcasing your males versus females control and males. And again, you see that same pattern with it, where in poly-IC you have more differentially expressed genes. This were the terms that came up. I am not a pathway. I do not look a lot into pathway, not my part, so I cannot tell you if there's anything super interesting for us besides the JAK-STAT pathway signaling. And again, this is interesting for us because this has to do with the estrogen prolactin pathway that you find in the MPOA. And again, you see the same pattern, but instead of having a flip-flop effect in the VMHBL, poly-IC either makes the sex differences stronger or it makes them weaker. For example, here you can see that in control, there is a little bit of a sex difference that goes away in treatment. Whereas here, the expression of these genes is pretty low. And then when you introduce treatment, it becomes more, it's higher. Same thing here. The expression changes. So whereas in the MPOA, you see a flip-flop exchange where poly-IC is actually changing the expression of the genes. In the BMHBL, it's just either increasing them or dampening them. So maternal immune activation may alter sex differential gene expression in adult MPOA. These were the overall findings. So in MPOA, we saw that there was a local relation between control and poly-IC conditions. Poly-IC seems to disrupt or reshape typical male to female expression patterns. And only a smaller subset genes show stable sex differences across both conditions. For the VMHBL, we found that sex differences in the VMHBL are robust and stable across conditions. And the poly-IC doesn't erase or flip the sex DEGs. It may slightly amplify or dampen them, but the males versus female pattern remains consistent. So the next question we wanted to answer was, are brain regions differences in gene expression altered by MIA? And the reason we wanted to look into this was because of this paper. So this paper looked into human data. They had tissue from control brains and tissue from autism brain, and they compare tissue from the cortical region to the subcortical region for both groups. And what they found is that in autistic brain, the DGs, the genes that were differentially expressed, decreases by a lot. So in control, they found 510 genes. In autism, there's only 8 genes. We wanted to see if we can find something similar in our data. And what we found is that instead of having poly-IC decrease DEGs, it created more DEGs. More DEGs came up once you introduce poly-IC. And why we want to do this? Well, although the MPOA and VMHBL are subcortical regions, the MPOA integrates sensory and social cues, and it drives motivation. The VMHBL executes behavioral output, mating, aggression, social approach, and together they have a circuit that regulates social and motivated behavior. Obviously, there's 3 more brain regions around the same area, but I just made the connection that MPOA and VMHBL have directly. So for this, what we did was comparing the sex differences, comparing the MPOA with the VMHBL, and seeing if there was any differentially expressed genes, but also how poly-IC changed this. And again, you can see that for females, the number of DGs increased. For males, it also increased. And overall, when we compare all control versus all treatment animals, the DGs also increase. Okay, so here you can see that regional differences in MPOA and BMHBL. There's regional differences in MPOA and BMHBL instead of sex differences. This figure shows a very tight correlation between control and poly-IC. Genes shared with both dominate, so the orange dots dominate. This means that the transcriptomal identity that distinguish MPOA and BMHBL is largely conserved regardless of any immune challenges. And this part is pretty preliminary data. This is something that we just found last week. And again, when we look into the cake analysis for the regions differences, we saw that for control, these are the genes that were MPOA, the terms that were MPOA skew, and these were the terms that were BMHBL skew. Whereas for poly-IC, these were the terms that were MPOA skew and BMHBL skew. Most of the terms stays the same, but some of the terms that are found in control dropped in poly-IC group, and new terms came up. This one, no, we're not gonna go there. So when we look to see if brain regions differences, if there's any brain regions differences in gene expression are altered by MIA, with the preliminary data, we can say that MIA may increase regional differences, but nothing more for now. So our overall finding is, one, we observe region and sex-specific vulnerability to MIA. MPOA and BMHBL respond differently to poly-IC. Females show greater region-dependent changes, and the MPOA is more transcriptomically sensitive to poly-IC. And two, there's gene expression patterns that suggest that MIA may rebalance typical sex differences. And many genes stay sex biased after MIA, where others flip or shift. And this is just why this is matter, the gap in knowledge and what we show. So most MIA studies do not examine both sexes. We did, and because of this, we found sex-dependent transcriptomal sensitivity. And regional specificity is poorly understood and studied. We compared two subcortical regions and found how MIA perturbs region-specific gene expression. And yeah, just my little acknowledgement table to the lab, my committee, and my undergrads, my current undergrads. Yeah, that's all. Questions?
About the flip-flop effect you were mentioning, did you look into more like what those genes are, if they're enriched for certain types of?
Yes, so let me go back, because actually some of those genes that no. So yes, I've been looking a lot more into those genes that are showing that flip-flop effect. And actually, well, here it shifted, but sorry. These genes right here, prolactin, so the PRL, PRLR, SOX2, and CISH. That's prolactin receptor, SOX2, and CISH. They're all from the same pathway, that prolactin estrogen pathway. And that has to do with that JAK2 pathway also. So most of the genes that are showing that flip-flop effect in the MPOA have to do a lot with the part of the MPOA that deals with the hormones release of estrogen, or how it regulates estrogen and prolactin.
any pathways involving ribosomes or translation being affected?
Not really. For us, I think that at least for us, like the most important things that we look into were like the cytokines to cytokine receptor, because we do introduce an immune response, the JAK-STAT signaling pathway, because it has to do with the whole hormonal response and like release of estrogen and prolactin, and neuroactive ligand receptor. Um, this is really preliminary, but I can explain a little bit. So this is for that last preliminary thing. And here we see more of that autoimmune thyroid disease, a lot to do with cells development, but also a lot more to do with the hormone pathway signaling being affected. So yes and no. In certain comparisons, we do see some cells, like some terms, regardless of it being affected, but most of the times we're seeing that pathway. It's just like, I believe it's that pathway, that something in that pathway is what's making all of this come up, but we have not looked in depth yet to see exactly what is causing that. Yeah.
So for your EEG,
Mhm.
where do you think they're coming from? Do you think it's like a cell composition change, or that the same cells may express, but they're expressing different?
That's a hard question because we did look into the cell percentage. We wanted to see if there was a difference in cell percentage between groups, and we didn't find it, right? And I also added the cell percentage, so like oligodendrocytes and neurons and microglia and all of that into the analysis at some point to see if that shifted the results, the DEG results, and it didn't. So I don't think it has to do with cells. Yes.
I'm wondering if you know what sort of effects, if any, there might usually be of immune activation on hormone signaling pathways just in adults and especially women. I'm wondering, like, you know, would we expect any parallel effects in the mother? Like, could this be a regulatory difference that is part of the general immune response, like in the mother, and then just has unintended side effects if some of the same regulators get into the developing fetus?
Yeah, so I've looked a little bit into it to understand just a little bit more of like what's happening, because we are thinking of moving some brain dissection early on to P1 to see if we can capture some of those maternal, some of those like early maternal response to MIA. And sadly, when we look into MIA studies, they really don't look a lot into the maternal side. If they look into the maternal side, they're looking more into like the placenta, so how their immune response is transferred to the placenta, but they do not look into hormonal effect. And then when we look into the offspring sides, a lot of the time it has been done in males, because it's really hard to, the way they explain it is that it's really hard to control for hormonal changes in females. It can be done. I mean, yes, it's harder, but we did it. And we hope that now for the next stage of the project, we're able to understand a little bit more about what's causing that. Because you believe that if it was a treatment effect, right? Something that came from the mom because of the mom response, you believe that it would also come up as a treatment effect in the animals, just not a sex difference that was introduced for poly-IC. So when we compare all control animals to all poly-IC, you would expect that something related to that would come up, right? But we didn't see that. But we can argue that actually what we're seeing is just completely normal too. Also, like, MIA work in relationship to neurodevelopmental disorder studies a lot of the cortical regions. They do not study a lot of the subcortical region. So even looking into brain regions like this, like the MPO and BMHPL, is something new that we did because they are highly sex differential. And we wanted to see if they have something to do with the sex disparity that we see in neurodevelopmental disorders. Okay, that's good.